Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. So hi Richa Yadav, a very good evening. So thank you so much for joining my session. So Dharani, hi, I'm good, thank you so much. Wishing you a very belated happy birthday. So Muskan, hi, hello. Hi Nishrag, hi, hello, hi. So Nishrag says a rainy good evening. So it's raining in your place. That's really nice. Yes, I'm good. Thank you so much. So come on guys, please do like, share and subscribe. So Ajay Sai, there is no rain in your place. Okay. So anyways, thank you so much for joining my session, all of you guys. So please do like, share and subscribe. So today we are talking about your plant physiology, especially your mineral nitrogen metabolism. Okay, so this bit is pending. So we are talking about this small portion. Yes, so with this chapter will be done. So on Tuesday we will start off with photosynthesis. So photosynthesis has a lot of huge equation, right? So we will start off with photosynthesis from Tuesday. So I have an announcement to make. So this evening at 6:30 we are having a quiz, and the quiz is from the examples of animal kingdom, especially chordates. Hi Shimpi, hello, a very happy good evening. Yes, I'm good, thank you. How are you? So please do like, share and subscribe. Okay, so that is the announcement. So 6:30 we are meeting again for a short menti where we are only talking about the examples of chordates from your animal kingdom. Okay, so shall we move forward with your mineral nutrition now? So that is nitrogen metabolism. So this is lecture 4 of your mineral nutrition. So this nitrogen metabolism is pretty important because this is how plants take up nitrogen. So plants need nitrogen in huge quantities, right? So if at all plants cannot take nitrogen, then what will happen? So nitrogen was the first element that we were talking about, right? So that is needed in greatest amount because it forms your backbone of your amino acid, your nucleic acid, etc. So if the plants cannot take up nitrogen, then what is going to help the plants take up nitrogen? So that is what we are going to see. So lecture 4 of mineral nutrition. So these were the topics we were talking about. So if you remember, we started off with the methods to study the requirements of plants. We started off with your hydroponics, right? So we started off with hydroponics. How in a defined media you can grow the plants without soil. So that was hydroponics. So based off for your hydroponics, if you want to have a defined nutrient media, we need to know which of the following are important. Okay. No, I haven't taught nitrogen cycles. I am yet to teach in ecology. I will be teaching on Monday. Okay. Yes. Fine. Okay, Dharani. Okay. So then based on this we have classified what are the essential minerals. So on what category did we categorize them? So we had 3 essentiality criteria based on which we categorize them, and the essential elements are divided into 2 parts. One is your micro and the other one is your macro. Hi Antonia B, a very happy good evening. So thank you so much for joining my session. So please do like, share and subscribe. Yes, so we had 2 types of essential element. One is micronutrient and the other one is macronutrient. So everything put together we have 17 essential elements which play a very important role. Now mechanism of absorption of elements, it is in 2-phased. Okay, so hi Satyendra, hello, after a very long time you are here. So where did you go in between? Are you fine? Yes, hope everything is good. So yes, good evening. So thank you so much for joining my session today. So please do like, share and subscribe. Yes, then mechanism of absorption of these elements. How are these elements absorbed? So these elements are absorbed by your apoplast, means by apoplast it is passive, and by your symplast, which was your active method. Yes. Okay, then translocation of solutes. How does it move through long distances? So you have your xylem vessels by which it moves. Then soil as a reservoir of essential elements. How soil acts as a function? Yes, so Shimpi is giving us the list. Yes, Nishrag says apoplast is passive. Yes. So here also we spoke about the toxicity. So this mechanism of absorption of elements. So toxicity of some element also inhibits the uptake of other elements. So that is why you have what is your critical concentration. If a mineral element decreases the dry weight of the tissue by 10%, then that is toxic to the plant cells. You have a base level concentration in by which you need or require the element. For example, so this is the optimum amount, that is critical concentration. So this is the concentration in which I need the element, okay. So above which, so if I have a concentration above this, then this leads to toxicity. So if I have the mineral present in a concentration below this, it leads to deficiency symptoms. Thus, toxicity of one metal can also lead to the deficiency symptom of other. Example is your manganese. So, your increase in manganese, they actually inhibit the uptake of your iron, calcium, etc. Yes, Ravi Ranjan Shah. Yes, a very happy good evening. So, thank you so much for joining my session. So, come on guys, please do like, share and subscribe. Yes, Anthony, you're right. So, you're giving me the list of macronutrient. So, Mohit, Jai Hind, okay. Yes, hello Jai Hind. Thank you so much for joining my session. So, please do like, share and subscribe. So, these are all the points that we were discussing. So, we also discussed about the functions of the soil, and the only portion that is remaining is metabolism of nitrogen. We also discussed about each of the elements in detail. Your calcium, your magnesium, your boron, your manganese, your copper, everything we have discussed, and hope you understood whatever was taught. So, now the last portion, that is metabolism of nitrogen. Why is it a separate topic? Why is it being a separate topic? So why it is not coming under the usual mineral elements? Because some plants cannot absorb nitrogen on its own. So that is the reason. So here let's see. So yes, I have already given the recap. Okay. So metabolism of nitrogen, so talking about the nitrogen cycle. So nitrogen is the constituent of your amino acid, of your proteins, your hormones, your chlorophyll and many vitamins. So this is where nitrogen is being used. So once again, it is a component of your amino acids, because you have an amino terminal, you have a carboxy terminal, you have a H group and you have an R group, right? Everything attached to your alpha carbon. So then you have proteins. So you have peptide bonds where your amino acids are linked together. So again there is an NH present. So nitrogen is present. Then your hormones. So you have peptide hormones like your insulin is a peptide hormone. Then you have chlorophyll. The center portion, in chlorophyll you have porphyrin ring, which is made up of nitrogen, and many vitamins which have your FMN, etc. So they are also made up of your nitrogen. So plants compete with microbes for limited nitrogen which are available in soil. So be it a component that is required by plants, be it a component that is not required by plants. So everything is present in limited amount in the soil, and soil is a home to a lot of bacteria as well as plant roots. So even bacteria needs nitrogen, because once again in bacteria nitrogen forms the constituent of amino acid. So nowhere we have studied that bacteria does not have amino acid. Yes, bacteria does have the same biomacromolecules. So every living cell, every living tissue is made up of biomacromolecules, right? So plants compete with bacteria for limited nitrogen that is available in your soil. So nitrogen is the limiting nutrient, so that is in nature as well as in agricultural ecosystem. So in both the ecosystems, so nitrogen is a limiting nutrient. In the natural nutrient, so in the natural cycle also nitrogen is very limiting, and even in agricultural ecosystem, so talking about your plant base, so there also nitrogen becomes a limiting factor. Okay, so nitrogen exists as two nitrogen atoms. So why does microbe require nitrogen? So why does plant require nitrogen? Because nitrogen is the backbone of your amino acids and proteins. Of course, bacteria also has proteins. Bacteria also has central dogma of life happening, right? So yes, it also has proteins. Okay, so that is the reason it also needs nitrogen. So nitrogen exists as two nitrogen atoms which are held together by 3 bonds. So it is like this. So we write nitrogen like this. So it is held together by 3 strong covalent bonds. So this is how we write your nitrogen. The process of conversion of nitrogen to ammonia is your nitrogen fixation. So this conversion of nitrogen to ammonia is known as nitrogen fixation. Ma'am, why should we convert nitrogen to ammonia? Hold on, okay? Plant physiology kitne marks hai? Okay, so plant physiology, so each topic has a different weightage. So your transport in plants has a weightage of 1%. Your mineral nutrition again has a weightage of 1%. Photosynthesis has a weightage of 2%. And then respiration 1%, PGR 1%. So it is 6% weightage of your entire plant physiology. Okay, Sanjay Bhargav? Yes, so Shimpi is very cautious. So whenever I use sorry, she will let me know. Okay, fine. I won't say sorry. I will see. So it comes spontaneously. Yes, the process of conversion of nitrogen to ammonia is your nitrogen fixation. So from N2 we are converting into NH3. So NH3 is your ammonia and this is your nitrogen fixation. So in nature, lightning and UV, they provide enough energy to convert nitrogen to nitrogen oxides. So there is a problem that your plants cannot directly absorb nitrogen in the N2 form. Yes, so we need an alternative form, so we are trying to convert it. Okay. Okay, important chapter. Everything is important because each chapter in transport in plants also solute potential, water potential is important. Since your photosynthesis is of higher weightage, so we pay attention to photosynthesis because it has 2% weightage. But as such, all the chapters in plant physiology are also equally important. Yes. So in nature, your lightning and the UV rays, they provide energy to convert nitrogen into nitrogen oxides. So N2 into NO, NO2, N2O. Okay. Plant physiology is very difficult. Really? So we are done with transport in plants. Now we are talking about mineral nutrition. So is it important? No, it is a bit easy. It looks as though it is very confusing and it is very saturated, but it is not. So that is why we discuss it in an easy manner. So if you have not understood the first 2 chapters, so please go back and watch my lecture. And from Tuesday, we are starting with the next chapter, that is photosynthesis. So please join us for photosynthesis. Okay. So decomposition of organic nitrogen of dead plants and animals into ammonia is called as ammonification. So pay attention, if you have a dead rat, the dead rat also has amino acids, also has proteins. So nitrogen is stuck inside the dead rat. So in that case, the nitrogen has to be converted into ammonia before it enters the cycle. So that is known as ammonification. The decomposition of organic nitrogen of dead plants and animals into ammonia is ammonification. Okay? Yes? So some of this ammonia volatilizes and it re-enters the atmosphere. Most of it is converted into nitrate by the soil bacteria. So what are the process we read? So we read what is nitrogen fixation, then we read what is ammonification. So first we are talking about nitrification process. So let me make it clear whatever I was talking. So we are talking about your nitrogen cycle. Yes, so nitrogen exists in the form of N2, that is 2N with 3 covalent bonds in between. So nitrogen is converted into ammonia and this process is known as nitrogen fixation. Okay. So sometimes your nitrogen is also converted into oxides of nitrogen. So who provides the energy for this? So this energy is provided by UV rays as well as by lightning. So in your dead plants and animals, your dead plants and animals is your detritus. So from here, the nitrogen is decomposed to your ammonia once again, and this process is known as ammonification. So this ammonia reaches the atmosphere, or it can be converted into nitrate by soil bacteria. So this is the fate of the ammonia that comes out of dead plants and animals. Okay? Yes, you are right, Nishrat. So this is what I was talking about. So now let's talk about nitrification process. Ready? So whatever I told, I have explained. The next one is nitrification process. So atmospheric nitrogen, so you have nitrogen in the atmosphere, so you have carbon dioxide, oxygen as well as nitrogen in your atmosphere, right? Yes. So this biologically, so how is this being utilized? So some of the nitrogen is used for biological nitrogen fixation, some for industrial nitrogen fixation, and some electrical nitrogen fixation or by the process of denitrification. So biological nitrogen fixation and any fixation of nitrogen is first nitrogen is converted into ammonia. So electrical nitrogen fixation, that is with the help of your UV rays and your lightning, it is converted into your nitric oxides, right? So basically this is your nitrate. So the ammonia that you convert is first converted into nitrite and further into nitrate. So this nitrate is also used in the denitrification process. Okay, so your nitrate is the form that the plant uptakes. Okay, so plants can take up nitrogen in the form of your nitrate. So that is why first it is broken down into ammonia, because your bacteria can convert your nitrogen only from your ammonia form. Nitrogen oxides are acidic in nature. Yes, you're right. Okay, so the plant biomass, so from the plant the animals eat. So from animals, if the animal is dead, then you have ammonification. Again, it becomes ammonia here. So please understand, so plants take up nitrogen in the nitrate form. So for that is the main reason it is getting converted into ammonia, then your nitrite and then your nitrate. So that is your nitrification process. So nitrate thus formed is absorbed by plants and transported to your leaves. So plants cannot absorb nitrogen directly, so it converts into ammonia. This is known as nitrogen fixation, and from your ammonia it is converted into nitrite. So this is nitrite and then into nitrate. So these two steps are done by your bacteria. Okay, so from here the plant takes up and this nitrate reaches the leaves of the plants. Done? Is this much clear? So nitrate thus formed is absorbed by the plants and it is transported to the leaves. So in the leaf, it is reduced to form ammonia once again. It forms amine group of amino acids. So once it reaches the leaf, so your nitrate gets converted into ammonia. So this ammonia forms the amino group of the amino acids. Clear? Yes. Ammonia is highly toxic for plants. Yes, that is also perfectly right. Till this much it's clear. So nitrate present in the soil is reduced to nitrogen by your denitrification process. So the nitrate present in soil is reduced to nitrogen by your denitrification process. Okay. So the denitrification is done by Pseudomonas and Thiobacillus. So in your soil, the nitrate that is present, yes. So how it is? So atmospheric nitrogen is there, it is converted into ammonia, and from ammonia it is converted into nitrite and then nitrate in the soil. Now your plants can take up, right? So this nitrogen, it can either be converted, this nitrate can be converted into nitrogen in the soil itself, and it is done by your denitrification process, and that is done by Pseudomonas as well as Thiobacillus. These are bacteria. So Pseudomonas as well as Thiobacillus. Is biological and atmospheric nitrogen different from each other? So biological nitrogen fixation, that is nitrogen fixation that is consumable by your biological organism, is biological nitrogen fixation. Atmospheric nitrogen fixation is in the form that your atmosphere can take up the remaining nitrogen. So that is the difference between both. So biological nitrogen fixation, the form that is used by your biological organisms. So atmospheric nitrogen, the form that is taken up by your atmosphere. Okay, yes. Okay, so denitrification is done by Pseudomonas and Thiobacillus. So now we are talking about biological nitrogen fixation. So what is denitrification process? So the reverse process of your nitrate getting converted into nitrogen. So this is your denitrification process. So ideally you have your nitrogen from your atmosphere. So this is converted into ammonia, then into nitrite and nitrate. So this reverse process of nitrate into nitrogen is your denitrification process, and it is done by your Pseudomonas as well as your Thiobacillus. Okay. So biological nitrogen fixation, so the reduction of nitrogen to ammonia by living organisms is biological nitrogen fixation. So in the first slide only I told you, right? So the nitrogen getting converted to ammonia is biological nitrogen fixation. The same line is given here, okay? So the enzyme nitrogenase is capable of reducing nitrogen, and it is present in your prokaryotes. So this enzyme is not present in higher eukaryotes, but it is present in your prokaryotes. So such microbes which can fix nitrogen, they are called as nitrogen fixers. These bacteria are chemoheterotrophs, your Pseudomonas and your Thiobacillus. Yes, you're right. So nitrogen fixing microbes could be either free living or they can be in symbiotic relationship with the plant roots. So nitrogen fixing, and whether they are free living or they are symbiotic, I have the examples as well. So aerobic, so you have nitrogen fixers. So first one is aerobic, so in the presence of oxygen, so they can be free living. So example is Acetobacter and Beigernicia. Okay, so these are the two bacteria which are aerobic and which are free living. The next one is anaerobic. So anaerobic and free living. So this is Rhodospirillum. So this takes place in the absence of your oxygen. Then cyanobacteria, which is free living, is your Anobina and Nostac. So I want you to remember all the examples right here. Yes, good evening. So thank you so much for joining my session. So please do like, share and subscribe. Yes, good evening Shivam sir. So thank you for joining my session. So please do like, share and subscribe. The enzyme has iron molybdenum cluster. Yes, you're right. So you're talking about the nitrogenase enzyme. So it has iron molybdenum cluster. Yes, Muskan, perfectly right. Okay. Contagium vivum fluid is Beigernica. Okay, yes, you're right. Anthony Abhi is also right. Okay, so reduction of ammonia to nitrogen is biological nitrogen fixation. So how can they go about this process? Because of the presence of an enzyme known as nitrogenase, and these prokaryotes are known as nitrogen fixers. So these could be free living as well as symbiotic. So you should remember which is aerobic, which is anaerobic, and which is your cyanobacteria or your blue-green algae which fixes nitrogen. Okay. So there are several types that are known. So the most prominent relationship is your legume bacteria relationship, which can fix nitrogen. So the rod-shaped rhizobium, it has relationship with the roots of the legumes. So legumes include your alfalfa, your sweet clover, your sweet pea, your lentils, your garden pea, your broad bean, your clover, your beans, all are your legumes. So the common association on the roots that they form is your nodules. Okay, so the nodules are the small outgrowth on the roots. So microbe known as Frankia, so this also produces nitrogen fixing nodules on the root of your non-leguminous plant, example your Alanus. Okay. Yes, so Nishrag is giving us examples of leguminous plants. Okay, so is this clear? So what are the points we are discussing? So nitrogen is converted into ammonia. So this is biological nitrogen fixation. Right? Yes, who does this? Nitrogen fixers, and they have enzyme that is known as nitrogenase enzyme. So most of these bacteria, they are free living or they are symbiotic. So the most common relationship of your nitrogen is with your leguminous plants. So rhodospirillum, yes, or your rhizobium, they are with your leguminous plants. So with your non-leguminous plants, you have Frankia. So this fixes nitrogen in Alanus, which is a non-leguminous plant. Okay, so your nitrogen is converted to ammonia. So ammonia is converted into, this is nitrite, R-I-T-E, then into nitrate. Sometimes this can be converted back into nitrogen, and this is known as denitrification process. This is done by nitrifiers, that is your Pseudomonas and your Thiobacillus. Okay. So from your dead plants or animal, if nitrogen is converted back into ammonia, so that process is known as your ammonification process. So from your detritus, if your nitrogen is converted into ammonia, so this is known as ammonification process, because ammonia gets accumulated. This is ammonification process. Fine? Yes. So rhizobium and Frankia are free living in the soil. So rhizobium in your leguminous plants, Frankia in your non-leguminous plants like your Alanus. So they are free living in soil, but they live in symbiotic relationship to fix atmospheric nitrogen. So the central portion of these nodules are red or pink in color due to the presence of leg hemoglobin. So now we are talking about the nodule formation. So how does this nodule formation happen? Yes, so shall we talk about it? How does this? Detritus is humus, so detritus is not humus. So detritus is your dead organic plant or animal remains. So that is your detritus. Okay? Yes? Deritivore, I mean denitrification bacteria. Deritivore are the ones that feed on your dead organic matter, but here, yes, you're right, it is denitrification bacteria. Okay, Satyendra? Yes. So now what happens? So how does this happen? So you have your roots. So you would have seen like this. So these are your nodules. So even in root modification, we would have spoken about your nodulated roots. Yes? So diamide, hydrazine, ammonia. Yes, you're perfectly right. So before this, I guess I left one step. So for this ammonia to your nitrite, it is done by your nitrobacter. Yes, so this is done by one bacteria, nitrobacter, if I am right. And from this to ammonia nitrite, it is your nitrococcus. Yes, these are the two bacteria. I guess I left this portion in between. So did any picture come based on this? Okay, so this is Nitrosomonas and this is Nitrococcus. So please remember these two bacteria. This is Nitrosomonas. So Nitrosomonas, this is Nitrococcus and Nitrobacter. So this process is known as nitrification. So ammonia, nitrogen to ammonia, so this process is nitrogen fixation, and from your ammonia to your nitrite and then your nitrate, so this is known as nitrification. Yes, I have written opposite. I found out the first one is Nitrosomonas. From your ammonia to nitrite, it is converted by a Nitrosomonas, and from your nitrite to nitrate, it is Nitrococcus or your Nitrobacter. Okay, is this clear? So please remember this. I forgot to tell you this. So now coming here for your nodule formation. How much energy is required? Yes, we will talk about it. So we need as much as 8 ATP. For one molecule of nitrogen, we need 8 ATP. Okay, so nodulated roots. So how does it happen? So bacteria is present in the soil. So you have your roots. The outer layer of your roots is your epidermis. So epidermis of the root has small hair-like projections. So they are your root hairs. Right? Yes. So the bacteria attach to the root hairs like this. Okay. So once they attach to the root hairs, then they start going deep inside the root. Okay. Yes. So confusion in bacteria name. First remember samosa and then come to bacteria. So remember Nitrosomonas and then you have Nitrosococcus. Okay, so remember it that way. So yes comes first and then coccus and bacter comes later. Okay. Yes, so you have your bacteria and it is present in the soil. You have your roots. The outer layer of your root is your epidermis. So the epidermis consists of root hair. So first the bacteria attaches to the root hair. So once the bacteria attaches to the root hair, it has somehow gained access into the roots. Right? So now what will happen? It will cause an infection thread. So it will cause, so this is the layers of your root. So it will cause an infection thread like this. So through the infection thread, it will go deep down into the roots of the bacteria, okay, and they will get into the roots. For example, if they have to go deep down into the root, so whichever layer they want to go, they will use the infection thread and they will go inside the roots, and once into that region, then they will fold inward and form your nodules. So it will be somewhat like this. So the soil particles, you have your bacteria, you have your root hair. First, they will grow in number, they will multiply in huge number. Once they are sure that they are present in enough number, then the root hair curls inside so that they collect the bacteria. So the bacteria starts moving inside. Then they develop an infection thread and they move to the pericycle and the cortex region. Then they form mature nodules in this region. Okay, yes, like antline. So this is your mature nodule. Ma'am, what is this nodule? So nodules are round-like structures inside which nitrogen fixation happens. So this enzyme nitrogenase is present inside the nodule. The center portion of this nodule is pink in color because it has another substance that is known as leg hemoglobin. What is this leg hemoglobin? So let me make it very clear. This nitrogenase enzyme coded by the NIF gene. It is coded by the NIF gene. The NIF gene in bacteria codes for nitrogenase. This is very sensitive to oxygen. If oxygen is present, then this enzyme will not work. So thus your nitrogen cannot be fixed or converted into your nitrate form. So it becomes a huge problem. Thus you have to have a scavenger of oxygen. So whenever oxygen is present, because this is inside the root, obviously oxygen will be present because cells are dividing, right? Sorry. So you have a scavenger, that is your leg hemoglobin, which scavenges the oxygen quickly, thus letting your nitrogenase enzyme work. Okay, yes, good evening sir. So thank you so much for joining my session. So please do like, share and subscribe. So this is clear? Yes. So there is something called as leg hemoglobin which scavenges the incoming oxygen. Because your nitrogenase is sensitive to oxygen, so you have your leg hemoglobin and nitrogen fixation happens inside the root nodule. Is this much clear? Yes, any doubts? So should I repeat any particular point once again? So Shimpi, Nishrag, Anthony, Abhi, is it clear? Yes. So should I repeat any particular point? So can I proceed? Yes. Okay, so I have overwritten, so just give me a minute. So see how much of energy is needed. You have your nitrogenase enzyme. So the binding substrate, the binding of the substrate to the enzyme. So you have your substrate. Your substrate is nitrogen, and with triple covalent bond, it binds. Okay. Host membrane group, pink pigment. Yes, you're right. Okay. So the host provides the leg hemoglobin. Yes, that is what you're telling. Right. So then it binds. Once it binds, then you have reduction. So you have 2H plus that comes into picture. So it forms NH2. So you have NH and NH. That is how it forms. Okay, so you have reduction, then you have one more reduction, now it is NH2. So you have further reduction, so it forms NH3, NH3, the products release and the enzyme becomes free. Okay, so this is how the substrate binding and the product takes place. So what happens? Your nitrogen is getting converted into ammonia. So for one molecule of nitrogen, you need 8 ATP. So for 2 molecule of nitrogen, you need 16 ATP molecule. Yes, it is a very energy expensive process. Is this much clear? Okay. So nodule contains all necessary biochemical components. So it contains your nitrogenase enzyme as well as your hemoglobin, that is known as leg hemoglobin, leguminous hemoglobin. So the nitrogenase is an enzyme which has your molybdenum iron protein. So just like Muskan told us, so this catalyzes the conversion of atmospheric nitrogen to ammonia. So ammonia is the first stable product of nitrogen fixation. So this nitrogenase is highly sensitive to molecular oxygen, thus it requires anaerobic condition. Okay, NOD and NIF gene. So NOD gene is also there, NIF gene is also there. Okay, so the one that codes for your nitrogenase. So nodules have adaptations that ensure that the enzyme is protected. So thus it contains an oxygen scavenger, that is leg hemoglobin. So microbes are aerobes which are free living, can thus fix nitrogen, do nitrogen fixation, which is an anaerobic process. So ammonia synthesis is high input of energy. You need 8 ATP molecules for the production of each of the ammonia molecule. Thus for one nitrogen to get converted into ammonia, you need 8 ATP, and thus totally it is 16 ATP. NID, NIS, where is it? So it is NOD and NIF. So these are genes that codes for your nodule as well as for your nitrogenase enzyme. Okay. Okay, so this is the process of nitrogen fixation. I hope you understood once again. Please repeat it. Okay. So you have your nitrogen. So nitrogen is converted into ammonia. So this is the process of nitrogen fixation. So this happens inside the nodule. So the nodules are formed inside the root. So the nodules contain the enzyme nitrogenase as well as it contains your leg hemoglobin. So leg hemoglobin acts as a scavenger of oxygen because your nitrogenase needs an anaerobic condition. Okay, so that is why this is present. For one molecule of nitrogen to be converted into ammonia, you need as much as 8 ATP molecules have to be invested. So the plants invest 8 ATP. So the bacteria only provides your nitrogenase enzyme. Okay, so how are the nodules formed? The root has curl, then you have an infection thread. So the bacteria reach the pericycle or the cortex cell and then they form mature nodules there. Yes, so this nitrogenase enzyme has your molybdenum iron cluster. So the active site is made up of molybdenum and your iron. Is this clear? Yes. Okay, so now this nitrogen, this ammonia, what is the fate of this ammonia? Now how does plants utilize this ammonia? So Shimpi, is it clear? Did you understand? What am I talking about? Yes. So shall I move forward? Shimpi, clear? Okay, so what is the fate of ammonia? At physiological pH, ammonia is protonated to form your ammonium ion, that is NH4+. So at your normal physiological pH, ammonia is protonated to form your ammonium. So most plants assimilate nitrate as well as your ammonium ions. So some plants, what happens? They take up in your nitrate form. Okay, so you have your nitrogen. So this is converted into ammonia. So what happens to this ammonia? So either this ammonia is converted into nitrite or nitrate and then taken up by the plants. So this can be one form, or directly the ammonia can be taken up and at physiological pH it is protonated to form your ammonium ions. Okay, the plants can assimilate, store your nitrate as well as your ammonium ions, but your plants cannot take up ammonium ions because it is toxic and thus it cannot accumulate. Your plants can store. What is the meaning of assimilate? Assimilate is to collect, is to store, is to accumulate. That is the form of, that is the meaning of assimilate. Okay, so plants either take up in the form of ammonia, but in the plant's physiological pH, ammonia gets converted into ammonium ions, but ammonium ions is toxic to the plant, so the plant cannot store nitrogen in the ammonium ion form, but it can store in your nitrate form. So let's see what happens. So how the NH4 is used to synthesize amino acids in plants. So basically that is the need for nitrogen, right? So that we can make your amino acid. So one is your reductive amination. So your plants have ammonia. So in your plant pH, in the physiological pH of the plant, your ammonia is converted into ammonium ions, but your plants cannot store or accumulate ammonium ions because it is toxic. Okay, yes, we have read in excretory system, right? So ammonium is the most toxic excretory waste. Yes, and it needs a lot of water. So how does the plant convert the ammonium ions into amino acid? The first way is reductive amination. The second way is transamination. So what is reductive amination? So in reductive amination, so you have alpha ketoglutaric acid plus the ammonium ions, plus you invest energy in the form of NADPH and you use the enzyme glutamate dehydrogenase. Thus the amino acid glutamate is getting formed, plus water plus your NADPH. So this is reductive amination. So by this process amino acid is getting formed. So you have your alpha ketoglutaric acid which is formed by other metabolic process plus your ammonium ions plus your NADPH. So in the presence of glutamate dehydrogenase, it is converted into glutamate, okay, plus H2O. Yes, you are right, Satyendra. So you have legume urites. Yes, you are right. We are coming to that point. So this is one form where your ammonium ion is successfully getting converted into your glutamate. The next one is transamination. So the amino group of one amino acid and the amino group of a keto acid. So the amino group of one amino acid is exchanged with the amino group of a keto acid and thus producing an amino acid. So here you have an amino acid. So these are ways by which your ammonium ion is converted into your amino acid. Thus the plants do not have to accumulate or assimilate your NH4 plus ions. Clear? Yes. So these are the two ways. So in plants like your soya bean, you have nitrogen which is assimilated in the form of urites, because urites have more nitrogen content compared to your ammonium. Yes, transfer of amino group of amino acid to the keto group of your keto acid. So there should be one amino acid acceptor and one donator. So this takes place between the amino acid and a keto acid. So this is the amino group of the amino acid and we are talking about the keto group of a keto acid. So when there is an exchange, so it leads to the formation of amino acid. Okay, yes. So this is the fate of your ammonia. I hope you have understood metabolism of nitrogen. So in the next class, so before starting the new chapter, I will bring some questions. So once we are done with the questions, we will move forward with photosynthesis, and photosynthesis is the most important topic. So with this, we come to an end of this chapter, that is metabolism of nitrogen. So if you have genuinely understood the way I taught, so please do like, share and subscribe, and please be there for the 6:30 p.m. quiz as well. So it is from the examples of your coordinates. So it will be 15 questions or 20 questions for 15 seconds each. So it is just like a revision for you. Yes? Okay. Chalo. Fine. Bye. So glutamic acid plus oxaloacetic acid. Okay. So we come to that form later. Okay, Muskan? Yes, today we have Menti, so please be there for the Menti. So have a great evening, and this is me signing off. So bye bye and take care.